Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry faculty. Well, in this video we are going to discuss about another hematopoietic vitamin, that is B12. So let's start with the discussion. Just in my previous video I had discussed about the hematopoietic vitamins. One was B9, which plays a very important role as a constituent of 1-carbon metabolism. Now, second hematopoietic vitamin is B12. Now, this B12 is also known as cyanocobalamin, because if we look at its structure, it has 4 tetrapyrrole ring arranged, and in between corine rings arranged, and in between it has cyano group. I'll just show you the structure. It looks like this. You can see that. Right here in between it has CO. So it is cyanocobalamin, and obviously it's a water soluble vitamin, that also we know. And I have told you one very important thing. Besides being a hematopoietic vitamin, the other important feature of this is that it has methyl group also, and it has adenosyl group also attached to it. So I can say it has 2 forms. One is adenosyl B12 and second is methyl B12. Right, depending upon this, it is going to catalyze its function. Now, another important point to remember about this B12 is B9 and B12. They are the 2 hematopoietic vitamins which are dependent on one another on one reaction in the body, which we will be discussing now. Okay, and then it is also known as extrinsic factor of Castle. Extrinsic factor of Castle. Now, first of all, the most important point, the unique feature of B12 which distinguishes it from rest of the vitamins is that this is the only vitamin which is of animal origin. That is why those people who are strictly vegetarians, they are more prone to deficiency of B12. This is the most important feature. Okay, so only vitamin of animal origin. So vegans are prone to B12 deficiency. This is very important, right? Now coming over to this, how this vitamin is absorbed in the body. What are the various factors which play a role in the absorption of this vitamin? So now what happens is, this is always absorbed with a very special component, which is hepatocorrin. So what happens here is, initially when we say, because we are taking this vitamin from the diet, so the moment it goes inside, it combines with a protein. So cobalamin will combine with a protein. Right. So if we talk about the mouth or the salivary glands, what will happen? Hepatocorrin will be there, which will help in the transport. So hepatocorrin, basically hepatocorrin is the one which is required for the transport. So now what happens is, the moment when we are eating food, the moment it reaches the stomach, what happens? This protein part is taken over by pepsin, or you can say pepsinogen will take this protein component and then it will combine with hepatocorrin. hepatocorrin. Right. Now what happens is, now one very important thing to remember about B12 is its maximum absorption takes place in the ileum. This is very important. Okay. No doubt it starts absorbing from the mouth, but mouth mein you can say it is only 3 to 4%. The maximum absorption will be in the ileum. So what happens here is stomach, then it reaches the duodenum and then ileum. So what happens here is that ileum has a specific protein which is known as 𝗵𝗵𝗴𝗺. So what will happen is कि ileum is going to take cobalamin, right, with 𝗵𝗵𝗴𝗺. And what will happen is, along with intrinsic factor of 𝗲𝗃. Okay, so what happens here is this cobalamin, when it is combining with hepatocorrin, pancreatic enzymes will take will digest this hepatocorrin. Means you can say कि cobalamin will always require somewhere or the other some support. When we are taking it from the diet, the protein is there, protein which is digested by the pepsinogen enzyme in the stomach, and then later stages mein pancreatic enzyme is going to take this hepatocorrin, and then it will be with the help of intrinsic factor of 𝗲𝗃 accepted by 𝗵𝗵𝗴𝗺, and there it will be taken over by ileal cells. Now the question is, what are the biochemical functions of cobalamin? जैसे I told you कि cobalamin is existing in two forms, adenosylcobalamin and methylcobalamin. So what are the reactions in which it is used? Okay, and this is how it is absorbed. So the most important point which you have to remember here is that it is known as extrinsic factor of 𝗲𝗃, but intrinsic factor of 𝗲𝗃 is required for the absorption, and the maximum absorption takes place in the ileum, where they have a 𝗵𝗵𝗴𝗺 protein which accepts it. Okay. So now coming over to what are the biochemical functions. Now the most important reaction catalyzed by B12 is, first of all, conversion of propionyl coenzyme A to succinyl coenzyme A. This is a very important reaction. Conversion of propionyl coenzyme A to succinyl coenzyme A. Now what happens here is, from where this propionyl coenzyme A is coming? This is coming from the degradation of odd chain fatty acids. Degradation of odd chain fatty acids. So what happens is, this undergoes carboxylation, means there is attachment of carbon dioxide, and this propionyl coenzyme A is converted into methylmalonyl-CoA. This is a very important reaction. So what happens is, this propionyl CoA undergoes carboxylation, and we have already studied for carboxylation we require biotin. So biotin, and the enzyme is carboxylase, and this propionyl coenzyme A is converted into L-methylmalonyl-CoA. Then this L-methylmalonyl-CoA is in the presence of B12 and mutase is converted into succinyl coenzyme A. So the main reaction is this, in the presence of B12 and the enzyme is mutase. right? So I can say that vitamin B12 is required for the synthesis of succinyl coenzyme A, and this succinyl coenzyme A can enter in the synthesis of heme. And we know it is also an intermediate of TCA cycle. So we can say TCA cycle, or it can be converted into glucose. So this is first very, very important reaction. And second important reaction is conversion of homocysteine to methionine. Now, whenever we talk about the conversion of homocysteine to methionine, we know that there is a difference of only the methyl group. From where this methyl group comes, the donor of this methyl group is obviously folic acid, but B12 acts as a carrier or a coenzyme. So it is like this methyl B12 will donate its methyl group and will be converted into B12, and this methyl group will come from methyl THF, right, which will be converted into this. So ultimately I would say that this donor or methyl group is coming from, means B9 is the donor and B12 is the carrier. But without B12, this reaction is not possible, and the name of the enzyme here is methionine synthase. So this is how homocysteine is converted into methionine. So these two are the very, very important reactions in the body which require B12. Okay, one is, you can say, in propenyl coenzyme A to succinyl coenzyme A. And second is conversion of homocysteine to methionine. Rest, it is also a component of ribonucleotide reductase. Component of ribonucleotide reductase. So these are the three important reactions in which B12 is used. Now coming over to the deficiency manifestations of B12. Now deficiency manifestations. Now if we talk about this reaction, first reaction, if I say that B12 is absent or deficient. So if B12 is deficient, there will be increased concentration of methylmalonyl-CoA, okay, and this methylmalonyl-CoA will be excreted in urine, right? So exactly what happens is, in deficiency manifestations, this particular reaction is linked with two manifestations. One is methylmalonic aciduria, because what is happening here is this propenyl coenzyme A, which is converted into L-methylmalonyl-CoA and which is finally converted to succinyl coenzyme A in the presence of B12. The reaction is not there. So there will be increased concentration of this, there will be increased concentration of this. So whenever the concentration of methylmalonic acid increases, it leads to methylmalonic aciduria. And second important thing here is, it will lead to subacute axonal degeneration or subcombined axonal degeneration, or 𝗲𝐶AD. Why? Because here what is happening, increased concentration of this propenyl coenzyme A and methylmalonyl coenzyme A will lead to excessive myelin sheath breakdown. Okay, that is why it will lead to this. So I will just write it here also, that there will be increased concentration of propenyl CoA, right? Increased concentration of methylmalonyl-CoA, and it will lead to increased myelin sheath breakdown. And it will lead to posterior and lateral axonal degeneration. This is very important, right? Cell tracks. So overall it will lead to, that is why it is combined. Why? Because it is leading to posterior and lateral corticospiral tracts. So myelin sheath breakdown excessive will be there due to excessive accumulation of propenyl coenzyme A. So these two are the very, very important points. And then third one is, it leads to pernicious anemia. Right. And then these are the three main biochemical manifestations or deficiency manifestations of B12. It leads to pernicious anemia also. Now what are the factors which can lead to deficiency of this vitamin B12? This is also very, very important, because I told you that B12 is the only vitamin which is of animal origin, right? So what are the factors which will lead to this? So the factors leading to B12 deficiency. Right. The first one is whenever there is autoimmune gastritis or malabsorption, right, or autoimmune gastritis. Now there are so many reasons to it, but in general if we say autoimmune gastritis, when there is destruction of the intrinsic factor, because I told you the maximum absorption of vitamin B12 always takes place in the ileum. So it will lead to autoimmune gastritis, or you can say in Crohn's disease. Right. Then in ileal resection. So these are the three major points which can lead to the deficiency of B12. And fourth is obviously if you are taking a vegan diet. Vegan diet strictly. If you are not taking any supplement, B12 supplement, if you are strictly taking vegetarian diet, then you are also prone to B12 deficiency. So strictly. Then the last one is, what is the biochemical assessment, how we assess that a person is B12 deficient. So next is biochemical assessment. Right. So if we talk about the biochemical test, the most important one here is Schilling's test. In Schilling's test, we use a radioactive isotope cobalt-60. So radioactive isotope cobalt-60 is used. Isotope cobalt-60 is used for the detection. Second is obviously peripheral smear will be there. right? And third one is simple methylmalonate levels. Very, very important. Okay. And fourth one is homocysteinuria. And this is indirectly linked to folate trap, not directly, but it is indirectly linked to folate trap, because I told you that I'll just write separately folate trap mein. The main thing which we have to remember is that B9 is giving methyl group, but B12 is deficient. Okay, so B9 will continuously keep on giving the methyl group, but the donor is giving, but the carrier is absent. So obviously there will be increased concentration of homocysteine leading to homocysteinuria. Moreover, this B9 will not be available for other reactions. So megaloblastic anemia is also a characteristic feature, but it is linked to it indirectly. And the basic difference between B9 and B12 is that if a person is B9 deficient, the methylmalonate levels will be normal. But if a person is B12 deficient, definitely methylmalonic acid will be excreted in the urine of that patient, right? So this is the basic difference between B9 and B12. And if we talk about the features here, so the main features here, the neurological manifestations, if we talk about neurological manifestations, would be the most important one. There will be tingling sensation, very important factor. Tingling sensation will be there. Right? Tingling sensation in the toes, you can say. In the toes, in the fingers, and sometimes that numbness will also come. Sometimes numbness will also appear. Right? And there is, you can say ki motor neurons will also be affected here. Very, very important. Right? And this is the basic difference, I would say, in B12 and B6 deficiency. And if we talk about the tongue, here it is beefy red tongue. Beefy red tongue will be there. So these are some of the very, very important features of B12 deficiency. Right? And this is the only vitamin, just now I told you, which is of animal origin. And these are the only two hematopoietic vitamins which are a component of B complex. Right? And this is, suppose if I have to differentiate between B9 and B12, I can simply say that both are catalyzing one same reaction. But in B9, excretion of 𝗵 is there. B9 deficiency. Okay. And in B12 deficiency, excretion of methylmalonate is there. Which distinguishes them from each other. Rest, megaloblastic anemia is the feature of both, because one reaction which is being catalyzed by these is the same, but this is the basic difference, right? So this is all about the video on B12. That's it. Thank you so much.